Hello and welcome to the seventh episode of Microbiome Gazers. This is a platform which is brought to you by Bugspeaks, South Asia's first microbiome test. I am Dr Debojyoti Dhar, co-founder and director of Leucine Rich Bio, South Asia's first microbiome company. To all the people who are watching us live and to the attendees, just to give you a brief, Microbiome Gazers is a platform which has been initiated by us to propagate, disseminate ideas on the microbiome. The whole idea is to bring in experts from various fields and discuss insights about this interesting domain, and so that every one of us, the researchers, the clinical community, and the students get benefited out of this. In this session, we will be talking about something which is very intriguing. Uh, the session that we have for today planned is on FMT, or fecal microbiota transplant. Now, to just give a brief, fecal microbiota transplantation basically is a treatment that involves administration of minimally manipulated microbial community from stool of a healthy donor into the patient's intestinal tract. And to talk about this very intriguing process, I have with me Dr. Siba Dolai from Sydney, Australia. Just to give you a brief introduction about Dr. Dolai, he is the principal scientist innovations at the Center for Digestive Diseases, the Center for Excellence in Advanced Gastroenterology in Sydney, Australia. He also heads the Research and Development Division of Accent Medical, a boutique R&D facility which uses cutting-edge next-generation technologies to uncover science behind microbiome therapeutics. So with this, I welcome Dr. Siba on this particular platform, and I'll hand over the microphone to Dr. Siba so that he can share a few words about himself. Off to you, Dr. Siba.
Thanks, Deb. I hope you can hear me. Is everything clear now?
Yes.
Apologies to everyone who has joined in. We had a bit of internet problems and other issues, but I hope you're able to hear me clear now. It's a pleasure and an honor to be able to speak on this platform. I think a very good afternoon to everyone who's joining in from India. And if there is someone from outside, evening or night, it's pretty much 9 o'clock in Sydney. Thanks for the opportunity to speak on this topic. I prepared a few slides, so if I may share that, and let me start off the discussion that way, and then we can take it from there. What do you think, Dave?
Before you start, I'll just give a brief on FMT to the general audience. FMT, or fecal microbiota transplantation, basically is it's an age-old process which probably got initiated in China, and in I think fourth century is what the earliest records suggest. And then, very interestingly, in World War II, troops in North Africa, German troops in North Africa, were made to eat camel poop to take care of their dysentery. And then in, I think, 1989, Professor Borodi, which the expert in FMT in Australia, where you currently work, showed that FMT can actually work for the first time in non-infectious diseases, because FMT is generally approved treatment for C. difficile infections. But now a lot of other non-infectious diseases are also being looked at. So with this small anecdote, let's start the session. So off to you, Dr. Dolai. You can start sharing the screen.
Thank you very much. Because because you started with the 4th century and the 16th century, I want to show you some very nice pictures of the people, because I was looking at today like who actually did that. So if I may share my screen.
Yeah. So that's the arrow button below.
Yeah, I clicked that. Share your screen. Yes. So can you guys see my screen now?
Yeah.
Okay. So from the Western perspective, what Dev was telling. So this is the main guys. So in the 4th century BC, I think it was from the Dongjing dynasty. Um, he was the first guy, Yi Hong, who did some fecal microbiota transplant, and then in the 16th century, it was Li Shen who did the fecal microbiota transplant, and they were mainly trying to get some kind of gastritis out, and that was the main field they were trying to target, right? But if you see before that, if I may go back.
You can put it in full screen.
So is it visible now to you guys?
Yeah, it is visible. You can put it on full screen.
Okay, now is it full screen?
But it's fine. It's, you can see. You can go ahead.
Yeah. So what I wanted to say, the first, which was the cases that were recorded mainly in Western medicine, was mainly from Eisman in 1958. He reported like 4 cases of enterocolitis, right, that he cured. And then in 1958 to 89, and more cases started to come into play. And I think that's when Professor Brody, who is my mentor and my current boss here, he was very involved in these fecal microbiota transplantation procedures here. And to date, they have completed around 38,000 FMTs, right, without any adverse reaction or any problem with those cases for fecal transplant. And I think after this, obviously the historical perspective, and mostly FMT was very effective in recurrent CDI infections. So if you can see the screen, so you see a healthy colon, and there is all the microbiota. And I think in these talks, there were lots of talks where you talked about the gut microbiota and how the amount of bacterial genes are much more than human genes, and we are more microbes than actual humans. So when there is a problem with these antibiotics, the microbiota is pretty much depleted, right? And then there is this C. diff, the Clostridium difficile spores, which are in there. They germinate, they will start having this big infection in the colitis, and again with the antibiotic treatment, you kill the active infection, the spores come back, and then it becomes more recurrent. And that's where the maximum utilization of FMT got its place. And even with a single dose, most of the infection was cleared. I want to show you one final slide on the recurrent CDI, which pretty much puts the stage for today's discussion. Is this paper in 2013, which was the first randomized clinical trial, and you can see a clear distinction of how infusion with the fecal donors.
Your slide is not visible. If you can, you can just point out on the slide which you are trying to show. It will be good. What we can see is we can see the right. Yeah, correct.
Good?
Yeah, this particular slide. Yeah, correct.
Yes. So is it good? So I wanted to say that the classical paper in 2013, which mainly laid the pathway for a proper use of fecal microbiota, right? This was a randomized trial where they used vancomycin with bowel lavage, and you can see a clear distinction of how fecal microbiota transplantation had a tremendous effect and cure rate in the patients. So Dev, I'll be stopping. I'll stop this sharing now, and as we go, probably I'll bring up the screen again.
Okay. Sure. So, yeah, this is a good perspective, Dr. Dolai, on the FMT. And as we started our discussion that how it initiated in China, and then mid 50s, late 80s when Professor Borodi came into the picture, and then things have been going uphill with respect to the FMT as we know it today. Could you share, because now you are at the at the epicenter of the FMT, revolution, let me put it that way. If you could share some insights on the FMT with respect to the intestinal disease, the non-chronic aspect, C. difficile, that you have rightly showed some slides on, and I think FDA also recommends FMT as the treatment option for people with RCDI.
Yeah.
And what I've been reading is that it has a success rate of close to 90-95%, which is amazing. But
Yeah.
what about FMT with respect to non-chronic diseases, especially intestinal diseases like IBD and IBS? If you can share some insights on those, it will be great.
Yeah. That's a very good question there. So the cure rates, let's say if you go with IBS or IBD, let me put these as the acute infection, because when you talk about CDI, it's mainly the older people, the older generation that are affected by this. But with IBD and IBS, is most of the newer generations who are affected. There is fantastic results which we are seeing now with FMT in this IBS and IBD, and I think there was a meta-analysis done as well where you get close to 50-60% results, right? But unfortunately, you don't get a standard response from every one of them, right?
Yeah.
Because if you try to put the same cap that we put on CDI, it's somehow not applicable. Like how we had this drastic effect in CDI, because it was mainly a commensal which went down and all. You get amazing benefits with IBD and IBS patients with FMT, but it's not across the whole panel. You don't see like everyone you treat some percentage. You see people who get fantastic results by the second round, right? And some people keep struggling with it, because I think it's a multifactorial kind of thing. The immunity comes into effect. Probably, like if you take one kind of, let's say Crohn's or ulcerative colitis, the trigger factors are so much more. There are so much incompatibility as well with the donors, because there is somewhere this immune component that's coming to play too, right? So that's making a huge difference in the effects that you get in IBD and IBS, right? But it definitely has the benefits. But unfortunately, I would say even with so much amount of research money that is poured into this, we still can't plug in all the loopholes on why this is happening.
Absolutely. To your mind, you've just mentioned some of the factors, but what if you have to point out to a single factor which determines the success of FMT in let's say intestinal diseases like IBD, IBS? What is it? What is it to your mind is the single most important factor?
That's a very difficult question, right? Because we are pretty much moving towards. So instead of finding what's the single factor that will enable you to have a successful FMT, what we have seen, a single donor, which we term as super donor, especially when you go to a IBD, IBS kind of issue. Forget about CDI. That's a different comparison when we do with IBD and IBS. I feel it's the donor, and somehow it's the cocktail that's in that donor. So we have like two donors who are pretty much the super donors. We tried pooling our very healthy donors and trying in IBS, IBD patient. We didn't get that effect. But with this super donor, somehow there is some component that's in there. I wish we could have like many of these super donors. And whenever that transplant is done, there is an amazing benefit that you see in this IBS and IBD patients, right? And I think that's the concoction, I think it's the exact amount of short chain fatty acids, the exact amount of bile acid, the exact amount of microbes that are able to convert those primary bile acid to secondary bile acid. So it's like a very big concoction, yeah? So we did try to match it through the microbiome studies, like metagenomics, and we did discuss about, like how the diversity or whether it's just a bacteria that's doing all the difference. So at this stage, Deb, I would attribute to a particular donor, a super donor. We are in the process of personalized FMT as well. We are trying to make it more, to go to the next level, because like you said, like Professor Borodi is a pioneer in this area. He has his personal investment to make this through. And so we are heading towards a personalized FMT, right? Based on your gut, who is the donor who would fit you?
Very interesting. I mean, this is a topic where we can really go on and on and on. There are so many aspects to it. So, see, the thing, one of the aspects which you also raised is the bile acid, right? And we know, especially for Clostridium difficile infection, we know that the taurocolate or the primary bile acid is actually used to culture the that bacteria in the lab, whereas the secondary bile acids are inhibitory to it. So
Yes.
complexity is there in the microbial milieu that it is very difficult to pinpoint to one factor and then probably suggest what is going wrong or what is going right. Initially the thought was that the bacteria are doing everything, but now, as you would know, there are so much research that has come up which is pointing to bacteriophages.
Yeah.
I recently read a paper where they had filtered everything out, leaving the bacteriophages, and they transplanted bacteriophages, and they found that it could do the same trick as the complete milieu.
I think we're talking about the Ott paper, right? In Germany, Ott and co-authors, they did this fantastic. The interesting thing is that in that paper, they really took everything off.
Yeah.
They did a sterility test as well to prove that there is no bacteria in there.
Yeah.
They did the transplant. I think it was 5 patients that they did the transplant to, and and they get complete cure, right? And like you said, the phages, probably it's the phages. We don't know, right? But the unfortunate thing with that is we had spent quite a bit of our time to replicate that, right? It's really hard. We couldn't replicate it, to be very honest with you. Like that's the complexity it is. I know they had set a standard, and I'm still waiting for another paper from the Ott group to come out where they did actually like a large trial. So I don't know what went wrong there or what went right there, but I'm still a bit skeptical about that paper, and I can't attribute it just to bacteriophages, but something is there. I'm still working on that. And as we go further, I'll discuss a little bit on that topic as well.
Fungi. Again, there was one paper where they had shown that Candida albicans levels are quite different in the patient versus donor, and then they suggested that even the diversity, richness of fungi plays a role in the success of FMT. So again, there are so many unknowns, and I'm sure being in Professor Borodi's lab and all those places, you would be looking at all these aspects in much more detail. But let that be as it may. I would like to ask you a specific question on donor. What are the criteria on which, at least in your center, you select the donors for FMT?
So. That's a very interesting question that you asked there, and now I'll tell you that's where the standardization is also going. So for us, for example, I'll give you some numbers. We screen 1,000 people, like 1,000 people are screened, right? And the final result we get is maybe 3 people who are suitable donors for our. Obviously, the standards that Professor Borodi has put in for his donors are very high, but I'll say the minimum it starts. Let's say we have a questionnaire, so we have an entire team of around 8 to 10 people who manage just the donor, and they are all registered nurses who are trained on this. The first thing is that we do a telephonic questionnaire on your food habits, on your different practices, how healthy you eat, what exercises you do, what is your kind of work, because it is a huge impact, right? We can't have someone who is working in a fishery and working with shellfishes. We can't have that. So what happens out of that 1,000, 300 of them drop out just with this questionnaire. Yeah. The second one is a face-to-face questionnaire where we have to see the physical fitness of the person, right? Because it has because now there is a microbiome component in every part of your body, and there is a omics study on it as well. So that's the second. And then once you pass that, the number comes down to almost less than 50%, let's say around 300 of them, right? Then they go through the normal pathological tests. So these are for all the pathogens that you usually see in the gastric diseases. You have to go through your HIV, your blood tests. So pretty much you are very healthy. And out of that, people drop out, few more. And then the final ones that are there, they are actually screened for little bit of microbiome studies as metagenomics studies, and we do some key bacteria analysis from them as well. So earlier, Deb, what used to happen is everyone used to use donors in a different way. There are people, you'll see lots of internet noise about people using home donors, and like I used my friend. Those are very few success stories, but what happened, so what we are doing here is like very high rate of screening, but now in Australia at least, it's very standardized. So the Therapeutic Good Association, like in Australia, it's the FDA in Australia called TGA. So TGA has rolled out a standardized platform now for even the screening the donors and all. So that's the stage one, right? So we used to have proper labs like a PC2 lab with GMP-like processes to produce the final material. But now TGA has come in and given exact procedures for every step, including the donor selection. So I think that's one of the big caveats of FMT as well. It is not easy as it sounds, because the donor is the hero of this whole game.
Absolutely. When you talk about TGA's role, basically we're talking about regulator coming into the picture. Of course, FDA, as far as I know, there is not much approval of FMT for non-infectious disease except RCDI. How does TGA look into this? Are they very open to this, or how are you taking this forward in Australia at least?
So in Australia, we're very lucky. TGA lets us do off-label.
Okay.
We have, so we still have not, I think in US you still can do an off-label, but you need an individual IND for each of those applications. But in Australia, I think we don't have rules for off-label use yet, I would say. So with the recent TGA regulations that came out, that's in early this year. So in that year, we had got these enhanced procedures, but we still have the option to do an off-label. So we are having lots and lots of international patients who approach, because I think it's kind of seen as the last resort. So you must have read there that pretty much everyone is talking in every field. Did you try FMT? Like whether it be chemotherapy, like post chemotherapy treatment. And because my dream when I came, because I was pretty much in a cancer field, like I was telling you before, when I came here, I kind of thought that anyone who is going through an antibiotic treatment or an advanced chemotherapy treatment should have at least one FMT pill to regenerate itself, right? So as of now, I would say Australia is very clean at this stage, because I think they did look into our procedures. So we at Center for Digestive Diseases, we have completed around 38,000 cases without not one adverse event. And based on that, I think TGA was thinking it is still okay, for us to keep continuing. The future, I'm still not sure, because we can't have randomized trial for each and every aspect of it because of the funding issues as well. But as of now, Australia is pretty safe.
Another question that I have is with regard to the donor stool samples. Now we know that there are multiple ways of doing this procedure. The best, the easiest is to take the fresh stool and then transplant it, but that is not feasible most of the time. So then there are other alternatives, like you go to a poop bank, like an open biome in US, or you freeze your donor sample in your center.
Yeah.
So how do you deal with? I mean, you have specific infrastructure for keeping these stool samples, and what are the requirements for having those stool samples in a place? What are the requirements for a poop bank, for example? If you can shed some insights on that.
So it's extremely hard, the food banks, because the investments are huge compared to the users. Yeah, because yes, freezers, like the freeze dryers, the lyophilization instrument. We have like very high degree of biological cabinets, anaerobic chambers, and all that. So that's where most of the processing is done. So even if we say it's a fresh transplant, there is minimum. Obviously, there is the yuck factor. You're still handling someone's poo, right? So somehow that has to be eliminated in the process. And the easiest way to do that is when you go into a chamber. So you use some of the robotics, an anaerobic chamber. So once the sample comes in and gets into the chamber, so it becomes a sample after that, because you don't interact with it directly. And I think that's the main concern, because once you keep it out, how do you keep something anaerobic which came out from you, and then you can't handle it on the table? So the main infrastructure is those cabinets that we get in. So those are mainly anaerobically controlled. Then we need some of the robotics part as well, so that we don't do that thing manually. So I think that the investment is the first part is that how do you prevent your staff working with the sample and remove the yuck factor. So that's where the robotics come into hand, and the chambers come into hand. There is a huge array of freezers and fridges that are the main component because of the frozen stool. And the other thing is like quality control. So at every step, the samples are taken. So you need to have a flow cytometer specifically. We are also trying to get in more cell counters. So the kind of counters you use your blood cells. So we are developing something to use to count bacteria as a measure. So because there is no point on mishandling a sample and ending up in dead bacteria or dead microbes in it. So that's the quality control step that's involved. And obviously the frozen thing is there is a system. So it's mainly a quality system where everything is tracked right from the beginning till the end, like whose still it was it and how many hours, up to seconds. We have a deadline of around 4 hours where the sample is completely coming out as a product, and try to filter it as much as possible so that it's mainly like it's like a brown solution, removing all the unused material. So those are the kind of investments that has gone in, and I don't think it's so the poop banks. The main thing is the freezers and the stability of those freezers and how the things were handled after that. So the investments is a little bit on the higher side, and the support is very little. We don't see grants where you say that what's the benefit of having a poo bank, right? Until why don't you have it in a centralized fashion and let the government do it? That's another factor as well. But the good thing there that what we have started now, like this is one of the big visions of Dr. Barodi as well, that so he wanted that what if we have so much effort in getting a super donor, can we ship the capsule around the world instead of someone standardizing it again and all, because all the world. So we are involved in such a big collaborative environment as well, and so that's a good thing. So just that will obliterate the duplication of high investments in different places.
Talking about super donor, I think we have a question from Dr. Srinath. Is asking, is it their ethnic background that matters, or their food and nutrition? What makes a donor a super donor?
This is an amazing question. Yes. See, I from a personal point of view, I'll tell you, I always wanted to see what if we take a 35-year-old Australian male with their food style and their lifestyle, whatever they did, and they are passing all our criteria to be a donor, and that comes in and it goes to someone in India, right? And the second thing I want to see, like an Indian man who has gone through all the, the pollution or whatever it is, they are exposed to so many challenges, and their immune system would be like a huge different one? And can that be used someone in Australia who have very little exposure to any other contaminants, right? So as of now, we really don't know, to be very honest, that what makes someone a super donor. And I think it cannot be replicated as well. Like, for a very interesting thing is, we have someone who is very old, like around 55 or 60 years old, still having very good microbiota, levels. But that children, they don't have it. They tried, but they fail all the tests. So I think it's pretty much like a fingerprint, right? You have it, you have it. You can't match it. And I don't know. I think there are even studies where people try to create artificial food. So you just create like a material where all the microbiota in its whole component. I think you must have read about those studies where people have been trying to create artificial material, but somehow it doesn't match. It doesn't have that effect.
Siba, again regarding the donors and the donor poop, what is the amount of the stool per session that is probably needed for a successful FMT?
I think there is between 150 to 200 grams. Is that normal stool? The usual amount. That's like, it's like one donation which comes around that. So those are filtered and they are put into syringes, and usually I think there are. I would say that's the usual weight that we take in, because every sample is weighed, and we see that with the amount of poo, that doesn't make that much difference. I think a normal cutoff around 150 to 200 grams is good enough at this stage. I think the repeated delivery is more important than like a more delivery. You know what I mean? Because anyway, those patients have to go through a routine before they, you need to have a clear bowel before you do this, just to give enough space for engraftment of the amount of bacteria we are having. We have seen even in CDI cases where you have a couple of repeated FMTs compared to one large FMT. I think it's the repeated FMT that makes more, so small doses but repeatedly. That had an impact. Like, this is not published, and most of the things that we are doing is not published because we couldn't reach a p-value. So we need that many amount of patients who are like. There was a big discussions we had, like how much different is a fresh from frozen, right? We do have evidence. I can show you some of the slides that I put in as well, which will go as we go. You'll see there's very little difference. The amount of you lose in that frozen is very little, and usually you get the similar numbers if you do fresh or frozen. So in that matter, I think to answer your question, it's around those 200 grams.
Mm-hmm. How many volume-wise, if I can ask you, what is the volume per syringe? What is the volume that is transplanted? Because
so I think it is, if you do it, the different there are different modes of injection. Like usually, the nasojejunal tube. It's not a very pleasant experience for someone to go through that. It's mainly through the colonoscopes. That's the easiest, and that's why it's around those 300 ml. That like you say, 50 ml syringes are loaded, six of them. That's the usual one through a colonoscope. Retention enemas, like with our experience at our center, like Professor Brody feels like he is the clinician. I'm his just the scientific part of it, and so we keep all the measurements. So my job is the measurements and to support him when he's doing. And I think it's around 300 ml that's usually used. Yeah, and through a colonoscope, and I think the results are hugely better when you go through a colonoscope than the other routes of administration.
My question for the frequency when you're doing multiple times FMT, do you use the same donor's poop the next time, or the donor can change?
So I think usually when we see that you are having that benefit, the thing is we try to match the same donor because you have already given a response, right? There is another concept as well for some people who don't have that apparent benefit come out. What we also do is we do a pool donation. So you get a mixture of donors, and there is some strategies. So these are like some of the stratification strategies that we take, that who is applicable, and sometimes people don't respond as well. So don't get me wrong that everyone comes in, walks out happy. People don't respond sometimes, and there are different strategies. Recently, I think a couple of years back, Professor Brody started having a pooled kind of donation where the different donors were mixed. That had a very good response as well. But I think as the regulations are coming in, it's getting much and much difficult. Like, it's getting more and more difficult to have a pooled kind of donation because of the quality controls. So that's the issue coming in as well. So now it's pretty much each donor to each patient, and once it's done, so usually for the ease of doing the FMTs, usually one of your closest kin is selected, like someone in your family or your close family, so that we can maintain that donor.
But I have a slight point to discuss on the close family part of donor selection. See, many of these, especially the infectious diseases, especially the intestinal diseases, there is a genetic component. For example, in IBD, the NOD2 gene, variants have been shown to be one of the, not say causative agents, but associated with this particular disease. Now, when you are taking related donor samples, there's a chance that the related donor samples also is coming from someone who has related, genetic profile. So therefore, in the long run, the same kind of milieu, although he might be healthy now, but what if there are certain because we are dealing with something which is very unknown. Of course, we are trying to figure out the constituents and stuff like that, but there are still a lot of unknowns in this domain. So this is my thought. Therefore, is although you are taking a donor from a relative who's probably very fine now, but he has a genetic component similar to the patient, and hence the microbial milieu might also have certain unknown things which can come up in later life of the recipient. So what is your thought on this particular aspect?
So there are two different sections. The first one is, we had recently completed one of the trials of around close to 400 people. In that, we have seen when you use a home donor. So home donor means mainly in IBS, IBD, the other acute intestinal problems. I feel that FMT is more of an adjuvant therapy to it. You know what I mean? It helps prolong your cure rather than cure itself. So that's one component that we gathered. There is a clear distinction of people, from the resolution of symptoms perspective, like how they change, the symptoms are getting better. We didn't see that difference. I know there might be a genetic component that's happening. We have patients who have been with us for like close to 25 years with that kind of thing. There's no major things that we saw that changed, like it was transmitted or something. See, like for example, in Crohn's disease, there is a component that is it microbacteria paratubically. There might be, so we are kind of hinting towards an infectious cause as well. So when it's an infectious, see, it's a hypothesis which has not been proved. So don't get me wrong in that, like, this is the only cause, but there is a component. We are having very big studies going on this because there is a therapy that's developed by Professor Brody, which is a combination of antibiotics for Crohn's disease. It's called an AMAT therapy. So he is a genius in having different combination therapies. He has proved in peptic ulcers and all, and even with Crohn's disease, one of the hypotheses is that maybe the causative organism is Microbacteria paratubically. and for that he has developed a triple therapy, and then FMT goes in, adds an adjuvant therapy. So once when the antibiotic course is over, you need an FMT to stabilize the situation, and we have seen some fantastic results from this. People have completely changed their Crohn's. They're very healthy human beings, but at some stages we also see there's no change at all. And that's why it cannot become the first line of therapy. But hopefully the next time I come to Microbiome Gazers, if you invite me, I'll say that, hey Dave, I found this thing and this is really working. But we are still working on it. To answer your question, no, we are not seeing any differences. Family is usually better for the logistical kind of view, like a son donating to the mother, and other close relatives. It's very easy to manage because we process the samples, but we get an active donor with us as well.
I think we discussed offline a long time back, which is post therapy, the importance of maintaining your nutrition. Because I think you gave me an example when we were discussing this offline about a gentleman who had his FMT done and he got cured of his disease, but then he started taking junk food,
Yeah.
and his problems resurfaced. So therefore, if you can broaden that particular thought and let the audience, whoever is watching us live, uh, get some take home messages for post FMT do's and don'ts.
Oh yes. So Deb, I was thinking I would not interrupt again with the share screen thing, but just for this one, I would like to share my screen just to show people that what's the impact of this. And currently, we are using FMT in a very novel but a problematic area called constipation. Right? We people think it's a lifestyle disease, and I don't think it's a lifestyle disease. After being with Professor Borodi, before that, I used to think it's a lifestyle disease. And you'll be surprised. We did a fecal transplant for a constipation patient, and I'm going to show you exactly something which will hit the nail in the head, like what has changed. And as I describe, I'll show you. I'll try to share my screen again. Can you see my screen?
Yeah, patient 108 constipation.
Yeah, patient 108 constipation, right? So can you see this is the first one is the donor, and you are very familiar. I think I'm telling this more for the audience who are here. The first one is donor, and this orange patch is pretty much Bacteroides. And so this is the base. So this is my constipation patient, right? Fully constipated, going very, very bad quality of life. And look at the amount of good bacteria, which we usually call Bacteroides, or the Firmicutes as well. Nothing. Just with week 1, you see it's starting to mimic the donor. So this patient got a fresh FMT transfer, and as the week progressed, sorry, Dave, can you see this?
If you could enlarge the screen, it will be good because
I think it is fully enlarged.
okay.
Can you see it now, Dave?
Yeah, it's similar. Okay.
Okay, let's go back.
Actually. Just now it got. If you can make it full screen or something, it will be easy to see.
Okay, I'm still having trouble with this. Okay, I'll come back to it.
Okay, fine. We can see it.
You can see it. Sorry, I'm not able to maneuver that. But
it's fine.
So this is the kind of thing I'm showing the patient. The first one, you can see my cursor moving around it. So in this is the first one is the donor, and this blue is completely the constipated patient, right? And once you do the transplant, Just with week 1, we can see the orange coming up, and the whole thing is the orange blob is pretty much bacteroides, and all the good bacteria coming back. And you see this patient is mimicking the donor now.
Yeah.
So this donor is fully fit and healthy donor, right?
Yeah.
And look at the progress up to week 28. The microbiome is pretty much reflecting what it was of the donor. And interestingly, on week 1, the constipation was gone. Right? So this is the power of what pretty much what Deb wants to hint. Okay, I'll just move this one. This is what I want to show Deb, that this is the impact that it has on new kind of conditions, right? And the diet becomes so important because up to week 26, the patient was on a diet which pretty much mimicked the donor, right? The good greens and like the beans and all that. It's amazing. You can see that clearly the constipation went away. The donor is pretty much now. That's the importance we are getting to see, like in autism, disorders as well. I was talking to you the other day. Right from week 2, you can see the changes, and after some days, the autistic features get reduced. I wouldn't say cure, but definitely. And constipation is beautiful, and I think now we never realized that there were so many cases. Like, it is a disease in itself now, I would say. It's a kind of new disease, and I think people who go through this and the effect it has on the mental disorders, brain fogs and all that. So now we're pretty much having a big queue of people who just want to get treatment for constipation.
In the last session of Microbiome Gazers, episode 6, we had Dr. Porwal. He's a proctologist in India, and he actually highlighted on the role of gut microbiota in constipation. And he gave a lot of interesting insights using clips from a movie called Piku, where constipation was one of the main background focuses. Siba, you put a very interesting thought, which is of use of FMT in autism spectrum disorder. We've got many clients who come to us on our website and say that can you guys do FMT for us for autism, which we can't, of course, because we are not there. So do you have any slides or any case studies that you want to share on FMT, or if you just want to say anything or any success stories that you want to share?
Yeah, so I did have a slide. It was similar to what we got for the constipation. So as of now, we have like a couple of, I'll tell you a case that happened last year where the kid was mainly presented because of a digestive disorder, right? So usually when you have an autism, you are somehow having a gastric issue with it as well. Obviously, it'll take another session just to explain the gut-brain relationship and all. But just in short, the patient had complained about lots of constipation. She cannot go to the toilet. I think she was a 14-year-old kid and cannot go to the toilet for up to 3 weeks. So the initial treatment that Professor Birdie was targeting was to clear off the constipation first, right? And because to clear that off, they realized that there's nothing left in it. How would she get the nutrition if there's all the bacteria cleared off, right? So then the FMT was done.
Okay.
And then what happened in 6 months? The mom calls back, saying that my daughter is able to say 1,000 words now, right? And so when we did analysis of we are not capable of assessing the autism thing. We don't work in that field, but we had our psychological psychiatric team as well who followed up that girl, and they said that from severe says come back to moderate, right? I think Arizona State University, there is a very big publication that came out very recently that talks about a randomized trial that was done on FMT and autistic kids. So we try and get novel solution from autistic kids. We offer that to international patients too as well. So what happens is there is a different kind of preparation that's prepared for them, probably in a milkshake, because it's very not through rectal route is very hard for kids. So we have a milkshake kind of preparation that our lab produces just for autistic kids, and it is phenomenal what is happening with them. But unfortunately, you can't take it as a cure. There are cases there is no improvement. Like I said, the problem with this is there is no black and white in this. Things work. Sometimes it is because of the donor. Sometimes you just respond very well, and not just you, your immunity and how you are built. And that's the reason that we cannot show a black and white story. But definitely, I would say huge. It was a big awakening for me personally, to see that this can be cured. And obviously, there is a huge advantage if you have a metagenomic profile done for it, like what you guys are doing an amazing job. And Dev has not paid me to tell that. And I'm just saying that there is the power of it. I would love to work with you guys more, because we don't have the time to do all of it. Because I would love to automate this process where I could completely be seeing these kind of profiles coming out day in and day out, right? But unfortunately, now the cost of FMT is prohibiting us for this. So all these metagenomic profiles that I'm showing, that is purely self-funded by Professor Brody, because we can't put it on the patient because it's already a burden on them. So I wish that grants could be starting to come this way, or maybe more and more investors should put money in your company probably, so that we can have more access to doing these things more often. I think the culture is still coming. People will try and get this out soon. Hopefully, let's see how it is going, because we all are doing it in a very small scale, because of the amount of funds. We have so many unanswered questions that need to be addressed. But
sure. Okay, so Siba, you have any more slides to show? Feel free to share the slides.
Yeah, I think that was the main one. I was just going to talk a little bit about the future of FMT. That's the last one, and showing it.
And I think we have a question also from Mr. Leander Corey. He's asking, what is the possibility of oral delivery, and share some light with regard to scale up of this technology?
Yes, I think that's the to end up. I'm not able to share this properly. Are you able to see this screen? I'm not able to enlarge the screen.
Yeah, it's okay. Yeah, we can see it.
Yes. So recently there was a publication just a couple of weeks where we had this maternal fecal microbiota transplant in cesarean babies. Yes. And this is like kind of a groundbreaking study that we always thought. Cesarean babies always had this problem of bacteria being transmitted from the mother's microbiome to the kid. And now, so this is kind of an oral formulation. We are doing lots of oral formulation to answer the question that came up in the panel. And that formulation happens in the form of capsules. So it is mainly a lyophilized product. So you don't have to really have a scope delivery all the time. What we can also do is develop capsules which can be taken on a daily basis as well. Patients who are on one capsule per day for an extended period of time, and they have got very good remission from their condition. So yes, and we have scaled it up as well. We're also launching a new GMP facility now to increase the production, because we got a lot of international clients who are interested in our capsule products. So if anyone is interested, I'm happy to share, like the details on how they can acquire these capsules or try to establish it in India. Definitely reach out to them, and they would contact me, and we can be in touch.
Great. Siba, my last question to you is, because we have time is up, so just wanted to ask the last question to you, which is, what are your suggestions to the clinicians or the doctors who are performing FMT or are planning to perform FMT in future? Any guidelines, insights?
I think the first, if you really want to see what's happening, the first thing is standardization. So whatever you do, it has to be standardized. That's my first request to any doctor who is doing it, because that's the difference. What we are doing, we are not seeing too many clinics doing it in Australia as well, because it's not very easy to standardize, because we are pretty much having a like my group is around 50 people. We are just involved in standardizing the product, right? So we have like different very highly qualified scientists who are involved in this. The second one is, even though it's a microbiota product, sterility is very important outside the product. You don't want to introduce a mold into your fecal transplant when you're preparing in a lab. So there is absolutely no second thought for good manufacturing practice. It cannot be compromised. You must have heard about recent cases where there is multi-drug resistance bacteria being transmitted to a patient in US. That was a very sad story, but the reason we are having very little adverse event is we are taking care of all of this. It is only to protect the name of Professor Birdie, who was involved in so much. We can't have one bad case. So if you are a clinician, you're trying to establish it, definitely think of having an independent, even if it's a small, it has to be an independent lab that performs for you. It cannot be in the same hospital environment, and probably standardize the product, quality control of your product. Then obviously, we have learned so much, and we are always happy to share, and to collaborate. So I would say that standardization and quality is very important, and some good manufacturing practices as well.
That is something which I think is lacking, because many of the doctors, for the love of it, maybe they get into it without doing the proper checks and balances that is required.
Yeah.
And as you said, there are so many steps that one needs to take care of before this transplantation can take place. But many of those steps are probably missed out by many of the clinicians. So with this, I thank you, Dr. Dolai, for sharing these insights, and for I know it's evening in Australia, so I'm not going to pull you more on this, and I hope to invite you again, where maybe we'll talk about FMT for a particular disease in specific, and we'll go much detail into that.
Absolutely. Absolutely.
And thank you once again, Dr. Dolai, for your time. And if there is any question from our audience, I will ask them to punch in those questions on the chat button. If there are none, there is any question coming, then we will end the session now. Yeah, so I don't see any more questions. Thank you, Dr. Dolai. And I hope to catch you offline later. Not today.
Yes.
I don't want you to be awake, but we'll be talking.
Yes. And I just want to say, reach out to everyone else that if you have any questions, please reach out to Deb. Deb and me are very close friends. We always chat around. We wanted to make something happen as well, and we are still trying to have a good collaboration. That process is on. And if you have any other questions, please reach out to Deb, and we can keep that flow going. Thanks, Deb. Thanks for inviting, and very good night to everyone.
Bye for now.
Thanks, Deb.